thank you for the introduction. Good afternoon, everybody. I'm going to talk about how can we apply genomics for species discrimination. My research is focusing on one family, which is Tephritidae, and why is it important to study these pests? 20% of the losses in agriculture are caused by arthropods. And specific, the family of Tephritidae, they infect the fruits because of part of the life cycle occurs inside the fruit and then cause damaging to different species. Sorry. And here I bring two examples to understand the importance of this problem. How can we measure this impact in monetary terms? So the second is very remarkable that only one species cost $2 billion for being eradicated in San Diego. So if we think about that, how can we prevent this to happen? So how can we avoid this problem? The first line of defense is the detection. So we need to know which species is trying to invade the place. And what is so difficult to identify, to differentiate one species to another? The first reason is because this family is very diverse with more than 5,000 described species. And the other reason is because most of the pests are classified inside species complexes. So that means that they share many morphological characteristics and the identification is very hard. Here are three examples of species that are classified as complexes, and they are considered as major pests. Here is the South American fruit fly, which is distributed in all the Americas. Oriental fruit fly, which is originally from Asia, but it invades other parts of the world, including Africa, Oceania, even the state of Hawaii in the United States. The other complex is from Southeast Asia and Oceania, which is the mango fruit fly. We can look at the external morphology of these individuals, and it's very hard to tell apart and say this individual belongs to this species and this other belongs to other species. And in this case, this can be a total different outcome, because the first one that is in your left is Anastrepha fraterculus, which is a complex, and it's a very dangerous pest. It's extremely polyphagous, and the other species that is in the middle, that is very similar by external morphology, is just a minor pest and can just infest a limited number of hosts. And if you think that regulatory agencies, most of the times, they just intercept some larvae and pupae, this problem is even harder to be resolved. So morphology can help to understand what is happening here. But we need to use some molecular tools for this task. And the first approach that we can think is using DNA barcoding, the classical using the gene CO1 of the mitochondrial DNA. And these two examples showed that this method is not suitable for discriminate the species complexes in these two genera. So what is our plan to solve this problem? First of all, we need to identify all of these complexes across the phylogeny, and then design a population genomics study to understand which forces are causing this problem, and then we can determine the boundaries of each species. This is excellent for academic purposes, but how can we apply this knowledge? So here are the markers that we are looking for. We need to find some SNPs that are fixed or nearly fixed in each species. And then we would have the information for all of the family, all of the complex in the family, and then we can develop a protocol for species identification. But in the reality, the problems that we need to face is that not always we have the best quality of DNA. So many of the collection occurs in isolated parts of the world, and then needs to be shipped to our lab and spend hours in ambient temperature because of regulation between countries, and then that sample suffers some kind of degradation. And the other problem is that the fly is per se small, so the amount of DNA that we can obtain is limited. And if we think that in some cases we need to preserve that individual as a voucher in the museum, we only have the legs to do the molecular work. So the first data that we need to generate is the reference genome. Here is our recipe for generate high-quality genome assemblies. One is using combining the strengths of two technologies. One is the long-read sequencing using PacBio, and the other is using Hi-C protocol and sequence those reads in the VD instrument. And here is what we can do with a good quality sample. This is a sample that we have the control since the collection until the end of the process, because it's in our lab colony. This is a heat map of the chromatin interactions captured by Hi-C. The intensity of the red color means more interaction, and it happens when these regions are physically closely in the same chromosome. So using this method, we can define the scaffolds that are in squares in blue, and also we can see the contigs that are in squares in green. And in this case, for this species, we expected 5 autosomes, and it's a male, so we expected an X chromosome and a Y chromosome. And we have the assembly in only one piece for 4 out of the 5 chromosomes. So this is a really good quality genome assembly. But not all species can be modeled. So in this case, we use other samples shipped from Brazil in ambient temperature. And even this assembly is not as contiguous as the other one, we were able to place all the contigs in the chromosomes. And this is an assembly that we can use as a reference for population genomics. The other part of this plan is to use whole genome resequencing to study the variation that is happening inside the complexes. Here is the example that we are working on right now, which is the mango fruit fly pest. Here you can see in the red asterisks the members of the complex, and two of them were recently described at only one species, Bactrocera frauenfeldi and Albistrigata. Nowadays, it's just considered just one species. Just to have an idea, the taxonomic problems that occur in these complexes. We also include other closely related species. As you can see, even the species that are closely related, not part of the complex, are morphologically very similar. So we sampled across the distribution of this species from Southeast Asia and Oceania, Australia, and you can see that we have samples even from islands. And then these samples were deposited to the museum in the University of Hawaii, and then we selected some samples for this study. We extracted the DNA, and then we prepared the library using NEB kit, in this case, and then use ADEPT for making this compatible to the sequencer. And this is the expected output, in this case, from one flow cell. We expected the results in 48 hours, 300 gigabases of data, 1 billion of reads, and quality, 90% of the bases, more than Phred quality of 30. So this was the first time that I used this machine, so I prefer to be conservative and only select 10 samples per run. And we expected for those samples about a coverage of 50x. And here is what we obtained. The results were very consistent across the runs. We have more than 300 gigabases of data, more than 1 billion reads, and about 95% of the bases had quality more than 30, which is pretty impressive comparative with other technologies. We can see the graph of FastQC, that the quality in most of the cases was even more than Q40. And what we can do with all this high quality data? So in this case, we map the RIPs to the reference, and then we use deep variant to call the SNPs. In this case, we found 9 million of SNPs from those samples, and then we use just unlinked SNPs, in this case 2 million, to produce this phylogeny. This is a fully solved phylogeny and is consistent with other studies in this group. But what is the problem? The problem is that perform this analysis take too long. In this case, just one sample took about 12 hours for being for obtaining the SNPs. If we think about that we also need to infer the phylogeny, this could be days to be done. So how can we reduce this amount of data to have this response quicker? So in this case, we just select some SNPs randomly and then repeat this process 100 times and infer the phylogeny for each group of SNPs. And in 50% of the times, one of the species, Bactrocera frauenfeldi, wasn't solved. And in 25% of the times, the other species that is closely related to Bactrocera frauenfeldi was not formed a monophyletic group. And even when these SNPs solved the phylogeny as we expected, in most of the time, the support was very low. So the question is, how can we reduce this data looking just specifically for some SNPs that are highly differentiated in the genome? So in this case, we calculated the pairwise FST, which is a measure of genetic differentiation in windows of 50 kV across the genome. And this is the trend for this analysis in each chromosome. Each color represents this comparison for each pair of species. If we look closer to this result, we can see that in some cases we found peaks of FST or genetic differentiation in the genome. One of the possible answers or the explanations for that is that this one gene or a group of genes in this region has been evolved under divergent selection. If it's the case that we find some genes that are directly involved in the differentiation of this species, we can have the best markers for identifying this species. And if we just sample the best SNPs from this approach and then build a phylogeny and compare that with the reference phylogeny, we can see that even if this phylogeny was not fully solved, all of the individuals formed a group with a corresponding species. So these markers could be used as a protocol for discriminate these species. So how can we, what we conclude with this study is that even in the cases that we don't have the best quality of DNA, with the state-of-the-art technologies, we can obtain high-quality data. And in this case, we present a framework to identify regions that can be used for species discrimination. And then we find this group of SNPs that are suitable for discriminated species in the mango fruit fly complex. I want to say thank you to all the PBAR team that is in your left, and also the team for the University of Hawaii that helped me with the taxonomy of this. Thank you. Helped me with the taxonomy of this group.